All right, these are your chapter 14, section 3 and section 4 notes. We don't actually have to cover all of the material from section 4, but we do need to talk a little bit, especially on the types of disorders that are common in different ethnicities within humans. So we'll definitely get there. Before I go to section 3, though, I just wanted to make sure that we all understand how to use our rules of probability, because you're going to have a lot of different genetic crosses, and you don't always have to use a Punnett square, and especially if you're going to be working with these multi-character crosses where you're crossing 3, 4, 16 different traits. You do not want to do a Punnett square for that. You'll be working on that Punnett square all day. So I just want to make sure that you guys understand how to use the probability rule, whether you're using the multiplication rule or whether you're using the addition rule. In most cases, when I ask you to figure out a probability, I'm asking you to figure out the probability of multiple events happening together, right? So for instance, if we take, I'm just going to use this space that I have down here. If we take a parent who is big Y, little y, and big R, little r, and we cross that with a parent that is little y, little r, little r, and I ask you, okay, well, what is the probability of getting I can either do a phenotypic or a genotypic probability, right? So phenotypic, I could ask, what are the possibilities of getting yellow, which is the dominant trait, and round, which is the dominant trait of the second. So big Y is what codes for yellow, big R is what codes for round. So from that cross, what would be the probability of that happening? So we first just take a look at our individual genes. So when we're looking at our trait for seed color, this is our cross. We're going to be crossing a parent that is big Y, little y, times a parent that's little y. And there's several different ways you can do this. You can use your probability rules, you can multiply this out, or you can just picture, okay, what does that Punnett square look like, right? So if I picture this Punnett square really quickly, this is what it looks like. And I can picture from that Punnett square that I'm going to have 2 out of 4 boxes that carry that big Y dominant allele, right? So 1/2 of the time, I'm going to end up with yellow offspring, right? But I didn't just ask yellow, I asked yellow and round. So these are 2 independent events that I'm wanting to occur together. So we simply do the same thing with the second trait, right? Here's our second trait, big R, little r times little r, little r. If you picture that Punnett square, you're going to see something that is pretty much exactly like this one, only replacing the y's with r's. Right, so we're still going to have big R in 2 out of 4 of those boxes, right, or 1/2 the time. So the total probability of receiving yellow and round offspring from this cross, since I'm asking for 2 independent traits happening together, that is our multiplication rule, right? So 1/4 of the time, I'm going to end up with yellow and round offspring from that cross. Right. So there's several different types of there's limitless different probability problems that we can work through. The key here is that I just want you guys to understand how we're building these probabilities, and then when we would need to add probabilities together like what you see here, because there's multiple different ways of getting a particular outcome that you're being asked for. Right? So multiple different ways of getting at least 2 recessive traits. So we have to add all of the individual probabilities of 2 recessive traits being inherited together. If you're still having trouble or you just want some more practice with probabilities, look on our next Canvas page, and I've posted a resource there that has some practice problems. All right. Now with 14.3, we are going to get into the inheritance patterns that are more complex than what are called simple Mendelian genetics, or what we more commonly refer to as simple dominance. So there's a relationship between the genotype and phenotype that it sometimes can be simple dominance, as the pea plant characteristics that Mendel studied, but most heritable characteristics, whether you're talking about plants or animals, they're not determined by just one gene with two alleles. They're far more complex than that. But even with those complex genetic inheritance patterns that we're going to get into, the basic principles of segregation and independent assortment still apply. Because no matter how many genes, no matter how many different kinds of alleles, it's still true that we're talking about diploid organisms. So we have one chromosome that we inherited from our mother, one chromosome that we inherited from our father. They contain the genes for the same traits at the same loci. And when those chromosomes undergo segregation during anaphase of meiosis, we see those principles of segregation and independent assortment playing out even with these complex patterns of inheritance. So they still hold true, right? So the inheritance of characteristics by a single gene can deviate from Mendelian patterns when the alleles are not completely dominant or recessive, so they don't behave in the way that Mendel's genes that were dominant or recessive behaved. They behave in a slightly different fashion. Or sometimes we have a single gene that actually has more than two alleles. And then even further, we have identified some genes now that actually are capable of producing multiple different phenotypes. And there's a lot of reasons behind that. We're going to get into talking about each one. But we have identified that one gene is actually capable of making more than one product or more than one phenotype. So we have to talk about this degrees of dominance, and this especially comes into play when we're looking at a particular level of organization, right? Are we looking at the biochemical level? Are we looking at the genetic level? Are we looking at the organismal level? And one particular trait can change in its degrees of dominance depending on what level you're looking at. But the way that it is interacting, the way that the genotypes, the alleles are interacting determines their degrees of dominance. So we say that something is completely dominant, or sometimes you'll also see this referred to as simple dominance. Right? So complete or simple dominance occurs when phenotypes of the heterozygote and the dominant homozygote are exactly identical. And we saw this with Mendel's pea plants, right? So if the individual was big Y, or if they were big Y, little y, they were still yellow seeds. Right now, in incomplete dominance, the phenotype of the F1 hybrids is an intermediate phenotype, somewhere in between the phenotypes of the two parents. So we say that with incomplete dominance, we get this intermediate phenotype. Intermediate. So a great example of this is with snapdragons, a type of flower. There are red snapdragons and there are white snapdragons, and if you cross them with each other, they produce F1 hybrids that are pink, right? Pink is not red and it's not white. It's an intermediate. It's a mix. It falls somewhere in between, and it's a brand new phenotype, right? Because it's neither the dominant nor the recessive. So we call that incomplete dominance. And then codominance is when you basically have alleles that are equally dominant with each other. This is commonly seen in chickens. I raise chickens on my farm. So if I can, get them to stand still long enough, maybe I'll take a picture for you guys. But we have what are called barred chickens, which come from crossing black chickens with white chickens. And then you end up with these chickens that sort of look checkered, where half of their feathers are black and half of their feathers are white. So in the hybrid, or in that heterozygote, both of the alleles get expressed equally. It's not an intermediate phenotype. It's literally both of the original phenotypes being expressed equally. So those chickens have both black and white feathers. They're not gray, right? There's a difference there. So here's that example of our snapdragons. If we took that red snapdragon and crossed it with the white snapdragon, all of the F1 snapdragons end up looking pink because there's an intermediate. The dominant cannot completely hide the presence of the recessive allele, so we see this blend, this mix or this intermediate phenotype produced. But if we were to cross two pink flowers, then you'll see in this Punnett square at the bottom here that sometimes those red alleles come back together, so you'll make red snapdragons. Sometimes the white alleles come back together, so you would also get white snapdragons, and then about half of the offspring of a pink-pink cross would also end up being pink, right? So in the case of P shape, the dominant allele codes for an enzyme that converts unbranched forms of starch in the seed to branched form. And we learned back last semester that that branched form of those carbohydrates helps with the water balance, right? So if we have a whole lot of unbranched carbohydrates, that means we have way more solutes inside that cell, and that cell is that P is going to start to take on water. And when it takes on water, when that P ends up drying, it dries and it wrinkles, right? And that is what brings about this wrinkled trait, because in the recessive, there's a defective form of the enzyme that leads to an accumulation of unbranched starch. We never end up making things like cellulose or starch grains. They stay as, simple carbohydrates, and it causes a lot of water to enter the seed and then wrinkle as it dries. We see the same thing happening when we take a look at some of the recessive traits that affect humans. Tay-Sachs disease is actually a fatal disease that is also the result of a dysfunctional enzyme. In this case, the functional enzyme is responsible for breaking down certain classes of lipids from our diet. If we have the recessive allele does not code for a functional enzyme. It actually carries a mutation that causes the enzyme to be built improperly, and so that enzyme doesn't work, and those cells end up accumulating lipids, and this is especially true of brain cells. So at the organismal level, this allele is said to be recessive because it requires two of them. So a person has to be homozygous recessive in order to display or show Tay-Sachs disease. If they are heterozygous, they do not show the symptoms of Tay-Sachs disease. So at the organismal level, the allele is recessive, so it follows what we would call complete dominance. But if we look at the biochemical level, right, the level of the phenotype, right, it's then incompletely dominant because the dominant gene does actually make functional enzyme, but not as much as what is made in the homozygote, right? Because we only have one gene copy instead of two gene copies that are making those enzymes. But there's enough enzyme that's being made that prevents the symptoms from being displayed at the organismal level. So at the biochemical level, this genotype is actually acting as an incompletely dominant because it is forming an amount of enzyme that is intermediate between the homozygous dominant individual and the homozygous recessive individual. And then if we get even closer, if we get to the molecular level, the alleles are actually co-dominant because when you look at them, the dominant allele is expressing the functional enzyme and the recessive allele is expressing the non-functional enzyme. So we have equal expressions of gene product, which means that they would be acting co-dominantly. So this is just an example of how you can look at these different levels, organismal level, biochemical level, molecular level, and these genes behave in different ways depending on what level you're looking at, right? So the frequency of dominant alleles It's not necessarily that dominant alleles are more common in populations than recessive alleles. It's definitely dependent upon evolution. It's dependent upon the population in question, and it's dependent upon the environment, right? What would be considered to be the more beneficial, right, or the more advantageous alleles? Those typically are the alleles that are maintained within a population. So just to say that they're dominant doesn't necessarily mean that there are more of them. Right? For instance, one baby out of 400 in the United States is born with extra fingers and extra toes. And this condition, which is called polydactyly, is actually caused by a dominant allele. But it's found much less frequently in the population than the recessive allele. So just because an allele is dominant doesn't necessarily mean that it's common, right? Now, most of our genes in our population exist in more than two allelic forms. So when we were studying Mendel's pea plants, it was very simple. Each trait that he studied only had two forms. The seed was yellow or it was green, it was round or it was wrinkled, the flower was purple or it was white. But with complex organisms, like animals, most of our genes actually exist in what are called multiple allelic forms, where a single gene contains more than two alleles. A great example of this is our ABO blood group in humans. This gene is found on chromosome 9 in human beings, and it is actually governed by three alleles. They are called the I superscript A, I superscript B, and little I allele. And these three alleles end up determining what types of carbohydrates are present on the surface of our red blood cells. So in humans, we have A carbohydrates, we have B carbohydrates. Little known fact, there are actually several other classes of carbohydrates that cause far more rare blood types. So we do actually have other blood types other than A, B, AB, and O. But they're incredibly rare. So these are certainly the four most common blood types in humans. And it's because of these three alleles and how they interact. So the enzyme coded for by the IA allele is what adds the A carbohydrate to red blood cells. So if an individual has the IA allele, they will have the A carbohydrate. Same thing with the IB allele. The IB allele adds the B carbohydrate. So if they have the IB, then they will be blood type B. The enzyme that's coded by the little I allele doesn't add carbohydrates to the red blood cells, the surface of the red blood cells. So we don't have any of what we would call those antigens on the surface of our blood if we only have the little I allele. So we can do several different types of crosses with these alleles to see how they would interact with each other. The top chart here shows you each allele and what it codes for. Again, these carbohydrates that are being formed on the surface of these red blood cells are oftentimes referred to as, antigens. Okay, so this would be called the A antigen. Right, well, it looks kind of like a triangle. It doesn't really look like a triangle, but it's just a nice way of, graphically representing the A antigen. And then if you have the IB allele, you form the B antigen. And if you have the little I allele, you don't form any antigens. So looking at the possible genotypes that individuals can have and what their subsequent phenotypes would be, if you were IAIA, this would be homozygous dominant for carbohydrate A, or if you were heterozygous, IA little I, you're still going to make those A antigens because remember, anytime you have that IA allele, it forms the A antigen. Same thing if you are homozygous IB or heterozygous IB, right? If you have that IB allele, then you form the type B antigen. So individuals who are blood type AB become blood type AB because they have both of these alleles. They have the IA which forms the A antigen, and the IB, which forms the B antigen. And then individuals who are like me, and there's quite a few of us, this is actually one of the most common blood types in the world. It is called type O. And individuals who are type O have the two little I alleles, or what could be referred to as recessive alleles, because the only way for it to be truly expressed is to appear in a homozygous individual. So we do not make any antigens, and our red blood cells are clear of any antigens. So that's how we result with blood type A, B, AB, and blood type O. There is, I believe, I put an extra resource on next class page that will give you some extra practice in crossing individuals within these multiple allelic blood groups. Right, pleiotropy is when genes have multiple phenotypic effects. And this is far more common than we ever imagined even 20 years ago. So pleiotropic alleles are responsible for multiple symptoms of certain hereditary diseases like cystic fibrosis and sickle cell disease, and we're going to talk about both of those. So it's one gene that causes these particular hereditary diseases, but that one gene ends up producing multiple different symptoms, multiple different phenotypes from that one gene. So we call that pleiotropy. There's also some traits that can be determined by two or more genes. So we also have what's called polygenic inheritance. And this is where multiple genes can independently affect a single trait. We also have another category of two genes that are impacting a single trait that's a little different from polygenic inheritance, and instead, it's what's referred to as epistasis. And epistasis is where one gene affects the phenotype of another gene due to how their gene products interact. So let's go through a couple examples of epistasis and polygenic inheritance so that we can see the difference between these two. Starting with epistasis. In epistasis, the expression of a gene at one locus can alter the phenotypic expression of a gene at a second locus. So this frequently happens with coat colors in animals. This happens with Himalayan rabbits, and it also happens with Labrador retrievers, which are one of my favorite dogs. You guys know how I feel about dogs. Labrador retrievers come in three possible coat colors. One of their genes codes for either black fur or brown fur. Labrador retrievers with brown fur are called chocolate labs, and then with black, they're simply referred to as black labs. But there's another gene that, we can just use the alleles E, that determines whether or not pigment can actually be deposited in hair. So the first gene produces the pigment color. The second gene determines whether or not that color actually gets deposited into the hair follicle as that hair is growing. So if we don't have the gene for color deposition, it doesn't matter what the black or brown gene is doing, no color will be deposited. So here's what happens. If you have two heterozygous black labs, which means they're big B, little b, big E, little e. When they're mated, if this was strictly following Mendelian genetics, we would expect a 9:3 to 3:1 ratio, where we have 9 black labs, 3 black that are heterozygous, 3 black that are not, et cetera, et cetera. But if we actually take a look at what really happens with a cross of that nature, we actually end up with 9 black retrievers, 3 chocolate retrievers, and 4 yellow Labrador retrievers. So what's happening there is what happens with that epistatic gene, that E, that second gene. So notice here, when you don't have a dominant copy of that E gene, these particular individuals don't have the pigment, even if it's coding for pigment, that pigment doesn't get deposited into their coat, into their fur. So notice that in order to be a black lab, you have to have at least one big B. So these three individuals here should have been black Labradors, but because of the epistatic relationship with that second gene, their pigment didn't get deposited and they ended up as yellow labs. And this final Labrador down here was supposed to be a chocolate lab, but again, because of the epistatic relationship with that second gene, its pigment did not get deposited. So we end up with an altered, what's called an altered Mendelian ratio. Right, so anytime you end up with those altered Mendelian ratios, very frequently it's because of an epistatic gene, or it's simply because we have some more complex inheritance that's going on. Right, so let's compare that to polygenic inheritance. Quantitative characters are those that vary in the population along a continuum. Things like height, and eye color and hair color and skin color. All of these are great examples of quantitative characters. We don't have just 2 or 3 phenotypes. We have a whole continuum of phenotypes within those characters. And when we have that continuum, it usually indicates that there's a polygenic inheritance pattern at play. And typically what we see is we see this additive effect, right? 2 or more genes of a single phenotype can cause individuals to get taller and taller and taller, or darker and darker and darker in their skin tone, or have darker and darker hair, all sorts of things. So if we take a look at skin color in humans, skin color is actually controlled by many separately inherited genes. This is just an example. So this is really, really simplifying skin color in humans, but you can see here how if you don't inherit any of those dominant alleles, then you end up with very light skin tone. If you only inherit one dominant allele, you end up with a slightly darker skin tone. And as you continue to inherit more and more of those darker or more and more of those pigment alleles, you end up with darker and darker skin. So we see this sort of continuum, and there are far more shades than just the 6 that are illustrated here, but that's just to give you an idea, right? There's also this question about nature versus nurture, and I know you've probably heard this before. It is true that the environment can have an impact on phenotype. So this is another departure from Mendelian genetics. If you take a look at how the phenotypic character is affected by the environment of the individual. All right, so anytime you have traits that depend on multiple genes and as also combined with an environmental influence, we refer to these as multifactorial phenotypes. Okay, multifactorial. And a great example of this would be for plants. Sometimes the color of flowers will differ depending on what type of soil they're being grown in. So hydrangeas that are grown in basic soil tend to have bright reds, pinks, orange colors, and then if they're grown in acidic soil, they tend to turn bluish, purple, these more cool colors. So that's just one example of how the environment can influence phenotype. They are the exact same in terms of their genetic makeup, right? So there's no difference in genetic makeup between the hydrangeas on the left and the hydrangeas on the right. But because of their different environments, it caused different phenotypes to develop. So an organism's phenotype includes its physical appearance, its internal anatomy, its physiology, and its behavior. A lot of what we're going to focus on in class is the physical appearance because that's the easiest to observe. But I do want you guys to understand that our genes also determine our anatomy, physiology, and behavior. An organism's phenotype reflects its overall genotype and our unique environmental history. Right, so we do have several traits that follow Mendelian patterns of inheritance, but some that diverge. Right, humans, first of all, are not very good subjects for genetic research. Our generation time is far too long. Now our generation time is anywhere from 20 to 40 years. Parents produce relatively few offspring, and quite frankly, breeding experiments among humans are socially unacceptable. So we don't necessarily make good subjects for genetic research like the garden pea plant that Mendel used, but because we do have genetic research, we can still use basic Mendelian genetics to sort of track what has occurred across many generations in a single family. Right, so we're certainly not going to have breeding experiments in humans, but if we have an entire family to look at, we can build what's called a pedigree. And a pedigree is a family tree that describes the relationships between parents and children across multiple generations and the inheritance pattern of a particular trait. So what's important to understand is that every pedigree is only going to show one trait at a time. Now, my pedigree should look pretty similar in terms of its overall construction of circles and squares, right? So if we look here, we can see some of our very basic symbols that we use for pedigrees. If we use a square, that indicates a male. Circle indicates a female. If they're shaded in, they have the trait that we are following. If there's a horizontal line that joins a male and female, that indicates a mating. Back in my day, when I was your age, because it was ages ago, this used to be called the marriage line. Marriage. Marriage is what brings us together today. But now we've sort of, we don't call it the marriage line, we call it the mating line, because in present day society, not everybody does the whole marriage thing before they do the mating thing. So we're just like, all right, cool, we'll just call it mating. And then if that mating line is joined to a bracket of symbols like what you see here, so we would have a vertical line coming down like so, and it would have been attached to that mating line, then that indicates these are the offspring of those parents, right? And offspring are always ordered in their birth order. So this would be the eldest of their offspring, and she was a daughter. And then this is the second oldest. They then followed that daughter with 3 sons. Okay, so we can take a look at these pedigrees to get an idea of how particular traits are inherited. For instance, this first trait is a dominant trait that is being followed. So notice all of the individuals that are shaded in have a dominant big W that codes for what is called a widow's peak. A widow's peak is where your hairline comes down to a distinctive point on your forehead. So here is that boop, boop, widow's peak. Versus someone like Ms. Myers, I have a flat hairline, so I don't have a widow's peak. I would be little w, right? But we can trace the inheritance of that particular trait through a family by using a pedigree. So these are the symbols for the 2 daughters that we're looking at. This daughter who has the widow's peak, this is her symbol, and the daughter with the flat hairline, this is her symbol. So following back, we can see their parents, right? So here are their parents. And we can see that their father was the youngest of 4, and their mother was the oldest of 2, right? And then we can trace that back even further to these individuals' grandparents. So here we have the father's parents, and here we have their mother's parents, right? So we're going to do a little bit of practice with pedigrees next class. We're going to be practicing it to see if we can determine whether a trait that's being studied is dominant or is it recessive, and is it autosomal or is it sex-linked? So we're going to add a little bit to this discussion next class. So pedigrees can also be used to make some predictions about future offspring. We can use the multiplication and addition rules to predict probability of specific phenotypes, and we can give individuals some information about the likelihood that they may pass on genetic disorders. Right? Most of our genetic disorders are inherited in a recessive manner, which means that two recessive alleles must be inherited together in order to produce the disorder. These genetic disorders range from relatively mild or innocuous altogether to incredibly life-threatening. So recessively inherited disorders show up only if the individuals are homozygous for the allele, like Tay-Sachs, which we discussed earlier. However, individuals can be carriers. So if you have someone who is heterozygous, these individuals carry the recessive allele, but they're phenotypically normal. And again, that's very much like the Tay-Sachs example that we used, where the heterozygous individual has one functional copy of the gene, so they make the functional enzyme, which can break down the lipids and prevent Tay-Sachs disease. But the other allele is still dysfunctional, right? That's the carrier, that's the recessive allele. So most individuals with recessive disorders are born to carrier parents. Albinism is a great example of a recessive condition characterized by the lack of pigmentation in skin and hair. So you could have two normal toned parents, but if those recessive alleles come together in the child, little a, it will form what is called albinism, which just means that they have the allele that produces pigment, but much like us, it doesn't work. Or much like when we were talking about our Labrador retrievers, right? It doesn't get deposited, so we have individuals that lack pigmentation, and that is called albinism. If a recessive allele that causes a disease is rare, right, it's really unlikely that two carriers will meet and have children, and that even rare that the child would be born with the disorder. But this becomes far more likely if individuals are related. Right? If they're related, there's a higher chance that there are two carriers in that particular family, and we call these consanguineous matings between close relatives. Now, again, frowned upon in humans, but this is actually what we have done in a lot of our artificial selection over the years. So as we have bred different breeds of cats, different breeds of dog, different breeds of horse, different breeds of cattle, even down to our agricultural yields, corn, soybeans, we have done consanguineous matings in order to increase the chance of rare alleles coming out in the offspring. You know, people love little squishy-faced dogs like my two beauties, Webster and Bugsy, and those breeds of dog, pugs and Boston Terriers, were brought about through specific consanguineous matings to try to enhance very specific traits. So again, most societies and cultures do have laws or taboos against marriages between close relatives. But what's interesting is that the royal family lines in Europe oftentimes snubbed their nose at these taboos. They were very much about keeping the royal bloodline, if you will. So they frequently did mate within the family. And because of that, we saw several traits sort of borne out in very specific areas of the world because of their mating schemes, typically within the royal family lines. One of those is in cystic fibrosis. And cystic fibrosis is probably one of the most common lethal genetic diseases in the United States. It is found in about 1 of every 2,500 people of European descent. And the cystic fibrosis allele results, again, in a defective or absent protein. It's a chloride transport channel that's found in our plasma membranes that leads up to a buildup of chlorine ions outside the cell. So symptoms of this disease include mucus buildup, some of our internal organs, and abnormal absorption of nutrients in the small intestines. Sickle cell disease is another example of a genetic disorder with evolutionary implications, only this is found in African American populations. This disorder affects about 1 in 400, and it is the cause of a single substitution in one amino acid of the hemoglobin protein in red blood cells. So this is a type of gene mutation. In homozygous individuals, all of the hemoglobin ends up being sickled in shape. So it has an abnormal shape due to the accumulation of hemoglobin proteins in an abnormal structure. So symptoms of sickle cell include physical weakness, pain, organ damage, and in some cases paralysis. What's interesting about the evolutionary history of sickle cell is that heterozygotes, although they do have some sickling of their blood cells, they're usually healthy, all right? So they do tend to suffer some of the symptoms because, again, some of their red blood cells do sickle out in shape, so they sometimes do experience pain, lethargy, things of that nature. But it also turns out that heterozygous individuals are far less susceptible to contracting malaria than homozygous dominant individuals, people with normal hemoglobin. So we find that in African Americans, especially because of the high frequency of malaria on the African continent, that this allele has been maintained in the population. Even though sickle cell is lethal and it typically ends up killing the individual, we still have a high incidence of the sickle allele because of its ability to provide some immunity from malaria. So this diagram here just shows you the difference between a sickle-shaped protein hemoglobin. So here is that sickle-shaped red blood cell. And then if you have the normal allele, your red blood cells will be smooth. There is one particular dominant human disorder that we should talk about. Dominant alleles that can cause lethal diseases are very rare. They almost always arise by mutation, and there's really only one example of a lethal dominant in the human population that we have identified as of yet, because it actually is carried out in further generations because of how it progresses. A non-lethal dominant disorder is called achondroplasia. This is one form of dwarfism. There's actually several. But achondroplasia is a rare dominant allele. It is not lethal. It just causes different development of the skeleton. So people with achondroplasia tend to be far shorter than individuals of the normal phenotype. But there is one particular lethal dominant disorder called Huntington's disease that is a degenerative nervous system disorder. And what's interesting about this particular disorder is that it has no effects. It's not registered in individuals until they typically reach their late 30s to early 40s. And there's still a lot of research being done on Huntington's to try to understand the progression. But it takes that long to lead to the deterioration of the nervous system, so much that we would actually start to identify that the Huntington's allele is present. So the condition is unfortunately irreversible and fatal. But the interesting thing is that because it is not onset until that time, typically individuals have already gotten married and had children, and this is how the Huntington's allele is carried on to the next generation, because they end up having children before they realize that they have the allele themselves. Right, remember we talked about those multifactorial traits that are governed not only by several genes, but also by environmental factors. So we have loads of diseases: heart disease, cancer, alcoholism, mental illness, all that have been linked to not only genetic causes, but also environmental components. So no matter what our genotype is, the lifestyle that we choose for ourselves does have a tremendous effect on phenotype. So in some cases, if you know that you have a family history, you can seek some counseling from genetic counselors to get more information about your specific genes before you have children. But for the individual's sake, those lifestyle choices, and I know I tried to point out a lot of them in our Living a Life Cancer-Free activity, but making sure that you're eating healthy, that you're getting plenty of sleep at night, that you are limiting your exposure to carcinogens, limiting your exposure to blue light, limiting your exposure to ultraviolet radiation, right? There's all sorts of things that can make a huge impact on your resulting phenotypes. Right? So the end of chapter 14 talks about some of these counseling methods, some testing methods that you can go through in order to identify whether or not, a developing fetus has inherited any of these types of disorders. I'm going to let you guys read through that on your own because it's not necessarily part of our AP curriculum, but it is interesting. So if you're interested, take a look at that. And then at the very end of your notes, you have some of this review that just goes back into those descriptions and examples of the different types of inheritance patterns that we've talked about.